So before talking about chromatin structure and nuclear organization in the genome, we can talk about, or you can say that the genome actually, in every cell, is a kind of string of 2 meters long. But that string of 2 meters long is packed into the tiny cell nucleus and forms a three-dimensional network that not only is important for packing, has a structural role, but also is important to regulate the expression of genes, and in part cancer-related genes. So what we have done is we have analyzed the three-dimensional structure of the genome within the cell nucleus in normal visual differentiation and in CLL. And we identified first that roughly 30% of the three-dimensional folding in the genome changes during normal differentiation of the cells. And then the remaining 70% that is stable, we compared the normal cells versus the CLL cells. And we identified 350 regions that change the chromatin interactions and three-dimensional interactions specifically in the disease. And when we had a look at these regions, we identified that within those regions there were genes that are important for disease pathogenesis. Now, in terms of treatments, you always ask us, how is this affecting treatment? But there is one thing we need to clarify first, and it's like good treatments in the future will depend on very good, very detailed and profound biological research and basic research. So what we have done is we have for the first time explored the three-dimensional structure of the nucleus in CLL. So to talk about the treatment now, maybe we look, so this is like a first, we have opened that new layer. I can envision that if these CLL cells are kind of addicted to a particular 3D structure, using drugs modifying three-dimensional structure of the genome may be a therapeutic avenue for future, but not a near future, I would say, yes.